It's my great honor to be here to participate in this incredible conference. And also, I want to thank Marco for your invitation, and thanks also for your chair this session. And I want to thank you all to stay with me at the end of this presentation, this conference. Okay, I know if I finish, then you'll be finished for the conference. So I'll try not to take too much time. I have to make sure that to use the both pens here, okay? And this is for this. Maybe, I don't know. Okay, this is for sleepover, okay. As Marco said, I have joined upon my Drexel University in Philadelphia, so I don't talk that much. And Tsinghua University in Beijing, and my center is at the West Main Building. This is East Gate of Tsinghua. And in Tsinghua, I director institute, the biomanufacture, and also we have two laboratories. One is called Beijing Municipal Government, the key lab for biomanufacture. Another one is in Shenzhen. And we have a faculty member, we have quite a number of graduate students there. So we'll be happy to help you for your medical conference in China, okay? So, and I started talking about 3D printing, and now I want to follow those great talking this morning, and there's a lot of talk. And the 3D printing, we start from CAD model, we slice that, and we make a coffee mug. You know, of course, we don't just make coffee mug today. We make beautiful stuff here and very innovative products. And I'll show you architectures and arts and even cookies and even printing a hot dog, and they call it a sandwich. Okay? But for 3D printing, there are three key features actually people interested to use. One is innovation, another customization, and a rapid realization. It's because these three key features find a very niche application in biomedical field. So that's what we get into, we call the bio 3D printing. And imagine you have a small print machines, and with those nozzles, and those nozzles could be 20 microns, could be a couple hundred microns, could be like a droplet and for single cell, and could be continued, and you drop many kinds of, or many aggregate of the cells. Or you drop biomaterials, so you drop biomaterial with the cells. You put this small printer, we call 3D printer, into the clean room, or into cell hold, the culture hold, depends on the size. Then you build then line by line and layer by layer. And different color here could indicate you may manipulate with different material and different and growth factors, so on and so forth. So you build a three-dimensional model. And this three-dimensional model is something like this one. And for example, in this application, and we pattern endothelial cells and on surface of those structure, and we mix of fibroblast cell with biomaterials. And we have two kinds of biomaterials, the RGD modified biomaterial and regular and alginate. And we pattern this way, and we're hoping that RGD modified biomaterial release growth factor can attract those endothelial cell line up. And hopefully they form what we call the blood vessel. So to us, this is what we call regenerative medicine model, it's an angiogenesis model, angiogenesis model. And we're happy to say this model is not grew up from petri dishes, which is very difficult to get it. But we can use 3D printing system and to assembling cell in three-dimensional space and assembling biomaterial to make this what we call advanced biological model. So that's something we get interested to say, okay, then how about we focus on biomaterial, we focus on the cells, even proteins, and we give this simple definition here. We call bio 3D printing uses biomaterial, cells, proteins, or other biological compound as building block to 3D printing personalized, which is biomimic structure or in vitro functional biological model. And it needs to be driven by computer model. And this computer model is not randomly designed. And this computer model is, we call the bio model, is either according to patient's specific physiologic structure, for example, biomimic of the scaffold and the implants, or you design a cell microenvironment. So that's really want to have that, then all require the biologic functions. So use those information and in the computer model and driven cells and biomaterial to form what we call the 3D bioprinting. If we're looking for the development of the whole field of 3D bioprinting, we can trace back to 1995. That is the earliest of when people use rapid prototyping technology. Because as long as the technology become available, people interested to apply that for medical application. But in that situation, we're not that much required for implantable biomaterials. Basically, we're not even required for biocompatible because we're not put inside the body. We use this technology to printing those medical models for surgical planning, and it's some talk in the morning that cover for that. Then, and we use the biocompatible material. We're interesting to make permanent implants, for example, hip and artificial knees, and that's because the second level application with advances of tissue engineering. Then people need to use tissue scaffold. So this and the printing technology sure is really advantage because it's not restricted by those shapes, complicated geometry. Then people use biomaterial, but particularly degradable, absorbable biomaterial, and to make a complicated tissue scaffold. Okay, I have an example later to show. Then I put it as 2003 because first patent of the cell printing is about 2003. Thomas Boland has a patent in the inkjet. Later on, I'll show you we have multinozzle deposition. And for cell, that's also patented in the priority that start from 2003. So from that time, and people interested to use cell as biomaterial and to print it. So that's, we extending of 3D printing and also extending of the material usage into different domains. Okay, so we use cell as material and to build what we call in vitro biological model. For example, tissue constructs, disease, drug model, and I will share with you of this. But only become last few years, particularly, this few years, and this field become and more interested, get more attention. The funding will go in that direction as well. But in the level 1 to level 3, it's from the medical model to scaffold, and to my point of view, and I think those are the application stage, because technology is enabling. And people use is SLS, FDM, as SLA, conventional 3D print technology to make scaffold. We focused on for translational research, we focused on commercialization, and I'm happy to share with some application. I will be run very fast for time. For example, for Yao Ming, put here for the injury, and the doctor uses this to make a medical model to see what is the therapeutic method and how critical for the injury. And to make this the level of transplantation that people use the 3D printing to make a model to make sure that the connectivity of those blood vessels. And I work with Dr. Spengler for those particular disease, and we have a 3D printed model for electrode. We use CT, and the brain we use MRI, and we patent that, for example, and this for doctor to use to make the planning, how to design this electrode according to this mapping, and for surgical planning. And I work with the Children's Hospital of Philadelphia, and for this particular application here, it's like the cosmetic and also tissue engineering. And people use this technology to make implants, for example, this personalize the hip replacement and the knees and craniofacial. There's a lot of applications here. People use this for tissue engineering and make tissue scaffold, because we do need the scaffold to provide temporal 3D architecture, and for cell growth, and then people use the cells, get materials, and make tissue scaffold and put into either bioreactor or inside the body, and hoping that the functional tissue can restored. And a few examples are given here. For example, this is SLA, it's stereolithographic, but this is the macro scale SLA. And the people in the post-tech in Pohang University, they use this to make a smaller, about 70 micron, and also degradable polymer, and for the tissue scaffold. And I know Scott was here and he talked about this, but this his work when Suman Das in University of Michigan, this is used selective laser centering and to make this and scaffold bone replacement, and they even have a good functional animal model result to show. And the FDM people use that too. For example, the Hashimoto group, they published paper talking about FDM to make this about 250, 300, about 500 micron scale size of each strand is scaffold and for bone tissue gene application. And Theraform, an exclusively licensed MIT 3D printing, and in the develop this technology to printing of bone grafts as well. And Tsinghua University, and we also have what we call the low temperature deposition. We have a PLA, CTP, for example, mixed up, and we have multiple nozzle. Then we're printing this into about negative 30 degree C environment. Then we'll be able to produce those large pore for cell attached growth and also small pore. This is controllable from 5 micron to 50 micron. So in this way, and different hierarchy of the pore size that provide is space for the cell and also nutrition for the cell. And we get some good animal models results for that too. And we're extending this application to make blood vessel, for example, where we consider of 3 different layers and for the biomimic design and use different material. And this printing method to build those blood tubing. And we have some animal model to show, for example, in 6 weeks. No, this is 24 weeks, 6 months. We still have functional of the blood, the vessels here. And right now we're working on use this technology to develop a blood vessel within smaller than 6 millimeter. So that's ongoing research project working on. And also at Drexel University, we developed this very similar to FDM, but we don't use filament. We directly put the particle of the synthetic polymer inside, about 110 and 90 is for PCL. If for the high temperature of the polymer, we could reach to 300 degree and to mold it. Then through this heating the chamber here, then the filament and being deposited into this line, very much like FDM. And the deposition of this in the room temperature, we can have the cooling temperature as well if we want it. Then we have 3-dimensional, we build and many different patterns this intended for bones. And we even put into the small animal to show how the bone regenerated. Okay. But those for the scaffold application, if we use this printing the biomaterial for scaffold, we can see is advantageable because those technology enabling, and we can directly get into CTMR data, or we understand what the patient specific information, we build a model, and we can make this is actually try to mimic that the defect of the bone, and we can precisely fit in. So in that regarding, and is biomimic, is now restrict on the shape and hard control capability, and reproducible. Okay, so that's is quite important. The disadvantage for that is unless you use the microfabrication technology, if current SLA, SLS, or this FDM, it's difficult to produce those small pore size less than 100 micron. So if it's about the 200, 300 micron scale level, it's quite comfortable. Anything smaller than 100 micron, that's still a challenge. But of course, there is microfabrication, even nano 3D printing technology developed to address those issues here. Another thing here is it's difficult for you to put living cells inside because you do need temperature, you do need chemicals, and the harsh environment will kill the cells. So let's get into, then we say, okay, let's change the printing method, then we use cells material, okay? And when you use cells material, and of course, you have to have living cells, then you have the biomaterial, or you don't have the biomaterial. In most cases, you do need the biomaterial. Next slide will show why we need the biomaterial. But those biomaterial special is gel-like material and make cell not die. You mix them together, then this cell biomaterial solution delivered through the 3D printing technology to form a designed 3D architecture The architecture I showed before could be biomimic, could be simulant of the cell microenvironment, or some special biologic function model. You're seeding or printing cells simultaneously with construct of the scaffold. So that's a lot of people initially, that's purpose, and it's difficult to seeding cell into thick tissue scaffold. That's why people try to put it together. And also it controls cell density and spatial distribution, because virtually you are assembling cell in three-dimensional space. As long as the cable cell now die, you assign cell seedings, migrations, and incorporate hybrid scaffold material, different cells, big technologies available. And also it's consistent, automated, and reproducible. That's leaves the ground for the future of the SIGAP. You can make automated products. So that's the advantage. We do use gel. At this stage, we use natural hydrogel gels that we call the gel, and use this gel to protect the cell, because when you put the cell into these micro nozzles, into inject, those printing head, sometimes it get damaged. So those material encapsulate the cell could protect the cell, because that's the first function. And also the material transfer the cell into different places. So you transfer the cell as well. Thirdly, it's important, sometimes in order to keep the cell survive, get the nutrition in the thick of 3D structure, you need to build this grading structure, you need to build the porosities. And both sides, you do need this cell to provide this gel to provide stability. And ideally, even this is very much we need it. And we want this gel can provide the bioactivity too. Because go with cell and grow with cells. That's another challenge for those bio-ink development. That's people interested. And also, when we're printing those gel, most of this gel are very sensitive to the printing environment. For example, different concentration of the material you put, you get different viscosities. And also, at printing at different temperatures, for collagens, the matrigels, those gelatins, they are temperature dependent, those viscosity, and also time dependency. So those are very important. So in other words, it's not easy you mix up those gel with cell that you printed, because a lot of factors will affect on the quality, affect the cell viability. Later on, I will share some results for you that. And also, we are lack of the good gel. So we have agent gelatin catechins, and with printing collagen matrigel, it's very difficult to form the 3D structure. So there's a lot of development people need for this gel too. But if printing cell, and I put here maybe one of these four methods people use to printing. One is inkjet. That started from Professor Thomas Boland in the Columbus University, and change those HP printer and get rid of the ink, then put a cell with medium inside, then print it. And that's the first being reported. And later on, then, particularly for engineering, we don't like this HP because it leaves us very little to do. We don't want to redevelop those cartridges. So we say, okay, let's develop a printing head. But of course, there's another advantage for us to develop the printing head. Those printing head we call the bioprinting method, which you put cell material inside. These are force, and this force could be the air force, could be magnetic force, could be a screw-driven force, could be piston force, but push those material and cell pass through small nozzles. Nozzle could be, as I said, about 20 microns, a few hundred microns, and there's platform and movement to form three-dimensional structure. So this is quite a few and group working on to develop this, we call the bioprinting method. And the Dimitris group now put this into the real product, but there's acoustic vibration and the small passage here, and those vibrations and push of the cells come out through this outlet. And a few group working on use lasers, laser, the punch. You see this substrate here, and only substrate is a cell attached substrate. And laser move at a certain position and punch, give the energy, then punch those substrate out and form the pattern. Because it's laser direct writing. If you see directly printing cell, a pattern cell, and people might use just one of these four methods that they have. Recently, the few paper reported about that. There's a good paper report about printing cell application. For example, the post-tech group, and the printing those stem cells, and with inside into decellularized matrix to form a cartilage regeneration, because that's the story. And Professor Jennifer Lewis group and Harvard reported, this is advanced biomaterial, and reported they can form and printing this internal with collagen network and the printing channel and seeding into cellular cells and form blood vessel. And her group has another paper published in the Natural Technology to in relate to this part of work. And Dr. Atala's group and recently talked about the printing those cells to build this ear structure. Okay, we can see it here. So ear structure here. And then this is through printing method. And as I said before that we developed this technology, the animation I showed here is for this intended biological model. Actually, we have this pattern and patented early at 2003, the priority data. And we try to use different and pushing mechanisms, for example, what force, what kinds of nozzles, what material, gels, and what type of cells. And I would like to share with you one application we have. And of course, currently, we have a second generation of this printing system. Not only multi-nozzle, we incorporate with UV solidifications and directly make a solidified pattern. We do use this technology and work with NASA from 2005 to 2010, and for what we call the drug metabolism study. And what we do here is NASA interesting and for Mars landing. So those astronauts, they take medicine every day and for the anti-radiation drug, but NASA is worried, and those drug is good for the liver, but passes through liver, the metabolism of the drug, in other words, convert from EFC to HFC, how that impact to the secondary organ in the zero gravity environment. Because that is what is interesting. So this is like a multi-organ model. And it's difficult for them to have animal study put into space station. So that's why they come say, okay, and can we printing those, printing a liver, or printing a heart, printing a bone. And the most effort we put is to print a liver because we want to HFC come out from the liver and impact to the secondary organ. So that's the idea what we have. But of course, print a liver is not random. You just print something. We have to understand what is the liver and how we can say that this small macro organ structure sounds like a liver. So that's what we looking for the physiology of the liver. Then we find, okay, so those are hepatic cells, and this blood passes through here. So this is what we are biomimic designed and printed. So we hepatic cells into cells on the surface, and we let the medium pass, which is the drug of NASA's, that drug, and pass through here. Then we detect what happened here. And during this process, we think they have a metabolic process happened in this macro organ. And that's we're printing that we have all these process parameters, and I will not elaborate it. We have a few papers publishing this. And we're printing a small chip with about 1 centimeter and 1 centimeter, 750 micron of thickness, and to simulate a three-dimensional of those cell layout. And we do have data to show those 3D comparing with 2D dishes they have, and also and barrier of endothelial cells and impact to the heart cells inside under radiation, and dynamic and static culture here, and quite a lot of results that NASA get from those models. And we have 2 patents related to this. Okay, and we did not continue in 2010, and since I have joined appointment in Tsinghua, so we stopped this, but we're working on something else too. So in 2012, and under funding by the National Science Foundation in China, we have key research project. We're printing, this time is printing cancer cell, okay? Because we're printing those cells for regenerative medicine. Let's see how we can print a cancer cell to build a cancer model. This is another project we're working on. And we do have printing system in Tsinghua, is 2D, now 2 head, the printing head. And we use this to printing a 3-dimensional tumor structure. So, and our thought was that, and we want to build 3-dimensional in vitro tumor model. And we want to try to see what we can get from this 3D tumor model and compare with two-dimensional model, which people use frequently and for the cancer study. Okay? If any difference we get it, and we may be, then we see what the difference makes sense or not, then we know 2D model has a problem, because otherwise we don't have problem for the efficacy of the drug. So that's the idea. So we challenge the 2D model, and then we compare of the proliferations and morphologies and also MAP expressions, and compare those with 2D and 3D. And I put 3 biomaterials here, and that's indicate that and the complexity of printing those cells and build this structure, because you cannot just use AgNet and to have good structure, because you just have AgNet and those cancer cells will not give you a biomarker. So in other words, they are not function like cancer cell. You don't detect those biomarkers. So we have to figure out what is combination of gelatin and fibrin combined with AgNet together. As I said, we do need a structure and a bioactivity both together. And also, we need those to protect cell, we not let cell to die. If you put too much of AgNet, the viscosity too high, then the cell will die. But we work on with HeLa cell at that time. So that's we printing. Again, this is a grading structure. This grading structure allows us to keep it in 2 weeks at least, and inside we still be able to have nutrition pass through and keep the cell to survive. And we're carefully tuning those process parameters. And we have our cell printing about 94% the variable after printing. And we compare 2D and 3D and those morphology. And you have to forgive me to skip a lot of biological part due to time, but we have paper published. If you're interested, you can get into that. Or you want to talk with me after that. So I will not focus a lot in biology. But we do form this type of model, and our collaborator is a tumorologist. And the first biomarker he gave to us is, let's see if your cell can form a round and can form a ball. If you do, that's the first indication that's tumor, because only tumor cell and form like this. That's the way successfully we get it. Then we compare with proliferation, 2D and 3D. I only show you the difference here. We're not elaborating. We have 2D and 3D for gene expressions here, or now for MMP expression, and also for cone resistance and how the drug resistance for 2D and 3D model. And we do also gene chip and find the 350 of upregulated and about 670 for the downregulated. And some of those up and downregulated indicate the very important 3D structure and for the biomarker and compare with 2D. And we even push to the EMT for the cancer metastasis study. And we get some good data to show our 3D model and for the morphology-wise and for the protein histology and to show the indication of the 3D cancer model. So it's quite excited, and when I was asked, and I always tell that we have something, we have simple tumor-like model. It's not a real tumor model because we don't have blood vessel inside, okay? But those model, and we're hoping we introduce blood vessel, and we put multiple cells and looking in depth of communication. But even though we can quickly use this model and for personalized cancer treatment, for example, get this model cells for patient and build 3D model, then we test drug and test and how what's the therapeutic dosage, for example, and we can directly use that. So that is when we continue working on that. And recently we're also printing embryonic stem cell to work on for that. The idea for that is also we combine with existing method to see if we print in 3D those EB structure, we get better results. We know that embryonic cells will not function by itself. It had to formulate aggregate and those ball structure. So then the idea what we want to have here is, can we have the cell EB, embryonic stem cell, mixed with the hydrogel, with mixed of the cell, the gel-like structure, and let it grow into the big ball. And this ball, we use that for further differentiations here. And it's a few for your outcome I will share with. One is we'll be very careful to adjust the printing parameter. For example, for same process parameter, if we print HeLa cells, cancer cell, we can get 88% of the cell viability. But if we print in C12, C2 the cells, And we have about 55% variability. If we're printing those embryonic cells, stem cell, we have about 14% variability. So that's why the printing process parameter is very, very important. And you don't want to damage a lot of cells here. But we try to adjust it. We get also about 90%, 93% of the variability of cell, we can print it. And we have three interesting outcomes we'd like to share. One is, and the EB we formed through our structure is proliferated, is not aggregated. But if you compare other suspension and hanging up method, they aggregate EB cell to form ball. But in our case, after carefully study of this about 140 hours, then we find that our EB form is proliferate, not aggregated. That indicates that you have potential to form more EBs, and also you have potential to have much better EB, because later on we find that our EB form is 100% of retain what we call the pluripotency. So in other words, without any differentiation. That's another exciting news to see the EB to have here. And another thing is also we can adjust the culture time or bio-ink composition and to obtain different size of EB. Okay, that's the one. And also our population in our harvest of the EB is much higher than high up drop and suspension. For example, about 1,000 times higher than what they get it, and about 100 times higher suspension. These two methods are people use for get EB. Okay, and also we form much better the uniformity of EB through our method. This gives another indication for us to see that, okay, potentially we may be able to use those process or structure and through the printing to control the fat of this EB and form different sizes. And also we have all the different sizes EB put together, they may be potentially to differentiate a specific cell, but this is stem cell biology. But that maybe have one model you can differentiate into micro tissues. Okay, so that's another what we feel that quite the promising, and if you continue, and we work on that. And so those is something we're working using cell printing. And I would like to share with some challenges, as I said, is not you put cells, put material, and you print something which you can get it. First of all, you have to follow developed biology. So that is how the cells and tissue and organ and even structure being formed. So this is a fundamental rule, and we have to follow. And we do have a beautiful cell printing method because we use top-down, which engineering design model. Then we put those together. Then we use bottom-up and let it grow up together. Okay. So 3D cell printing is make 3D model and put in the middle of this development biology mapping, not start from very beginning. Start very beginning is petri dishes. Okay. So that is something. And first challenge, we know that. We know that as engineering, if it looks like an engine, it's an engine. But if the biology, you put 12 different cells, it looks like the heart. You have all the heart cell put inside, it will not be the heart because you don't have that cellular sequences. As a matter of fact, and I always said, don't expect that someday you can directly take from 3D cell printing system and print a heart and move that machine back and put that heart into somebody's chest and make the function. That may never happen. But you can printing structure then and give that cellular sequences and potentially grow as a heart. That is we have to follow, that develop biology. So that means that in the 3D, the cell printing, that whole process, we're very much interested in how we biomimics. Because that biomimics means we want to replicate cell microenvironment. We want to really get what we call biomimic structure. At least it looks like the structure we wanted. Second is we make sure those cells during the process environment that will not get damaged, variable, and very challenging. That is, we give the dynamic of those core cultures and all the microenvironment and we're hoping to get a function. So that's the whole way three. All these three we have to pay attention. It's not just printing out, okay? It's printing structure, and you armed for the functional of in vitro model you have. So that means that we have a lot of knowledge gap we need to fill because those are beyond development biology and petri dishes. Okay? In other words, biologists, they never have this printed model before. They always depend on petri dishes. So that is knowledge. But if we give them something like this, that is something we have to work together and to develop that field. Second, material. We call it bioink. Because the bioink is not just a cell delivery medium, and it's not just provide a structure function, it also need to provide a biofunction. So we like those bioink to go with cell, grow with cell, okay? And currently we have very limited availability for those. And third, the printing. The printing involves the machines and positions, and also how to control environment, and how we can reduce the damage of cells, and how we can 3D co-culture it. So in other words, this technology brings a lot of challenge and new issues, and in terms of engineering, and in terms of science, and in terms of technology. So but it's because of those challenges and get us interested. For example, we do have printing cell damage, and we can see the membrane break out and internal of nuclei be separated. Okay, it happens. It's just because of challenge and also brings opportunities. That's what we have here. For example, if you're looking for the involvement of tissue engineering, in 1998, the NASF working definition for tissue engineering is engineering life science and for functional tissue, because those are keywords. But in 2007, and the 12 different agents get together, they don't say the functional tissue. This is the use of physical, chemical, biological, and engineering process to control direct aggregate behavior of cells. If you do that, you're working on tissue science and engineering. It's much broad. With this broad of the definition, the field changes, not just functional tissue. It's regenerative medicine, it's disease model, it's drug model, and it's cell tissue on chip. All of this, and we deal with the cells. To my understanding, physical engineering process, even including the self-assembly of a biological process to control cell, will be related to the cell printing. Okay, so that's plays the role. And also, we do need a good drug test model. We all understand it takes at least 10 years, the $1.2 billion develop one new drug. And the reason is we have limited of the human trial model. We have different of the human with animal model, and we're limited for 2D model, people interested for the stimulated 3D models. And those 3D models probably be made through the 3D printing method. And for example, one of we use to make disease model is we want to use assembling cell method to build these cancers, multiple cell. One cancer cell with a healthy cell, and the number of cancer cell with number of healthy cell, fully cancerized cell. Then we try to understand, it's not me to try to understand, my collaborator try to understand how this cancer cell influence other cells to become cancer. But this type of model may be only being accomplished through 3D printing method, because that's what we call the biology model. So if back to my conclusion, the slides, our goal here is we are the engineering. We develop better tools or better model to help biologists to better study biology. But it's not just for biology, and also, of course, to help engineering to design, manufacture better medical device as well. Okay, so, and I thank you very much, and would like to answer any questions if you still have time. Yeah.
Well, you have amazing activity in this area. Thank you for sharing that with us. On a number of the slides, you mentioned applying for patents and receiving patents. Usually, that focus goes towards commercialization. And I'm wondering if you can share with us, have you been able to license patents or anything along those lines?
Okay, Good question. That is one part. That is one part I did not share with you. Yes, we are working on that. In 2004, I remember January 1st. Then the first thing I do is register a company because Drexel wanted me to have a company. Drexel even put some small money and for me to continue for that, and I also fortunate to get somebody interested, and for my patent at Drexel, and I spent $1 million. Sounds. Later on, I find that I'm just a professor. I don't know how to negotiate, but we do. It's $1 million, and we get an exclusive license. And for the company, Drexel does not share any of the royalty, so that is why. Okay, before if Drexel share 7% of the stock, it's only about $250,000 or less than 200,000, and give up that 7% of stock, and they ask for $1 million. And we did. We licensed that. We have a company, and that is one. And also right now we try to build another international company. That's a U.S. company. In China, China has a very, very interesting strategy because Chinese government, and they focused on the commercialization. When the government gives you a project, they ask you have to have a company, okay? So, and they find we have those cell cancer model project. They want to fund it. They don't fund it, but they said, okay, you have to have a company. So for that, then we have to form a company. So we do form a company and in Beijing. And the company, carry on the government project, and also the company and carry on to sell the printing device. And in both U.S. and China, and we have quite many years, let's say 15 years experience to printing biomaterial and to printing cell, and we want this being commercialized. And right now we have about a line up about $15 million, $15 million to form an international company, and to build this from bio-ink to bioprinting, and also leads to those advanced drug model organ chip, and for that commercialization. Yeah. Thank you. Yeah.
Hi, thanks for your talk. I got a question about the commercialization as well, but kind of more of a geography question. I heard you mention that you travel between US and China 20 times in a year. And so where would you say is the best, maybe not US or China, but in the world thinking about regulatory and competitive environment, assuming that bioprinting, the hardware side, people are able to figure out, scale mechanisms by which to produce living tissue and components, where do you think it would be the best geography in order to commercialize this technology for you? Would it be China? Would it be US? Would it be CE in Europe?
Europe, and it cannot be US, cannot be in China. Okay, China is even strict than the U.S. because they follow U.S. If you don't have in the U.S. release, they pretty much they don't do it. And I see that because I serve on the advisory board. I'm a committee member for regularity of the bio-3D printing in China, and so it's difficult. It's difficult. So, for example, commercialization for my product, any of the product, I don't get into implants. I only get in vitro model, but for drug, for machines, not trying to get implant. And for cell-related product, and unless except for the simple stem cells, which US already gave FDA approved, for any of the cell-related, 10 years, I think you don't expect you can get any approval in China. And I guess in the US, we do looking for example, we have those workshops for NES and NIH. FDA always participated, but I don't see any clearly roadmap for them to consider this as a product. Yeah, so though difficult, Europe is better. Yeah, EU market. Yeah, thank you. Any other questions? Okay. Okay. Thank you. Yeah. Thank you very much.